Good afternoon. Today's topic is about the non-infectious abortions. Why in the frame of animal nutrition? Well, because most of the agents causing non-infectious abortion are related to the feed and feeding. This topic has great practical and economical importance for breeding sows, but to illustrate the whole landscape, studies with other animals will be presented too. In the general study plan, we will start with the introduction. After we will see the spontaneous or habitual abortions caused by stress, lack of major nutrients, and mineral vitamin deficiencies will follow. Environmental and endogenous pollutants like toxins and heavy metals. Allergy immunoincompetence or competence. And finally, Legionella syndrome, algal toxins, etc. At the background, we can imagine every time troubles of the circadian rhythm, light-darkness ratio, changes in the body composition, background metabolic diseases of the mother like diabetes in human and troubles of the sugar and ketone metabolism in the animal kingdom, and genetic sensitivity. Our goal is a better reproductive performance and not only a simple prevention of the abortion. Before all, we have to review the definitions after the up-to-date Merck Veterinary Manual. What is the abortion? Abortion is the termination of the gestation after organogenesis completed, but before the expelled fetus could survive. It's very close to the embryonic death if the gestation ends before organogenesis and the fetus will be expelled. And the stillbirth means a death of a full-term fetus. Its lungs are not inflated at that time. Many etiologies of abortion may also cause stillbirth, mummification, weak or deformed neonates. In the frame of the introduction, we have to speak about the causes of the abortions. Basically, each factor causing death of embryo and or contractions of uterus may cause abortion. In this way, all the teratogenic chemicals can be considered as cause of the abortion because may kill the embryo and after provoking uterus contractions. In this way, as we have to see, the cancerogenic and teratogenic substances means also embryonal mortality, and by this way a secondary cause of the abortion. The data are many times retrospective. In a big breeding farm of breeding sows, you can only see the parameters at the end of the year that the average litter size decreased or the number of the newborn piglets decreased. In some place they can find also the clinical signs, but not typical. For the today's lecture is not in question the problems of nidation, the changes of uterine fluid pH and composition caused, for example, by protein overfeeding and etc. This is not abortion. As summarizing potential etiological factors, uh, all can be derived from one key factor just before the abortion. This is a surge of prostaglandin E2 in the mother's blood. And it can be provoked by stress, lack of nutrients, minerals, and vitamins, pollution, allergy, and immunocompetence, and toxic effects of microbes and algae. In the general study plan, we arrive to the spontaneous or habitual abortions caused by stress, and you see that it may have a series of different causes. The social stress is known for you from the laboratory animal science and bioethics. Calhoun used rats to modelizing the social stress, and by overpopulation, the rate of the abortion and the drop of the parental maternal care was significant. The pheromones, the so-called Bruce-Barton effect in mice and rats, is called in the scientific popularizing literature as abortion. This is not an abortion, this is only a prevention of the nidation, but you have to know about it. But we have to concentrate to the environmental stressors like the heat, the humidity, the state of the floor, noise, etc. The transport and the place in the hierarchy in sows and cows, etc. In this model trial, the effect of transport stress was studied on sex hormone blood level. After GnRH injection, they checked the plasma LH level with and without transport, and you see that consequently their susceptibility, potential abortion if they were gestant would be different. Yes. The change in social status, or in other words, a high and a low social status in the hierarchy, may cause a great stress. The decreased animals who lost their place in the hierarchy were compared with the animals, with the dairy cows who increased in the hierarchy scale. And you see that even the fine reproductive traits like calving to conception days, insemination per conception, milk yield, cell count, and lameness score are significantly different, and any time is worse in case of the low social status. Of course, that if these animals were gestant, the danger of abortion is also higher. In the general study plan, we arrived to the real nutritional factors, the lack of major nutrients, and the mineral vitamin deficiencies. The basically nutritional part of the etiology of abortion are the lack of major nutrients and the mineral vitamin deficiencies. You know that the major nutrients are the carbohydrates, the fat and oils, and the protein, and we can summarize them as energy. The energy deficiency mostly will prevent the fertilization, and the abortion is not typical owing to energy deficit. But in case of female goats, the abortion can be provoked by energy deficiency at about 110th day of the pregnancy, gestation. The case is the same for the protein deficiency. A real big deficiency will prevent the fertilization itself. From the minerals, many of a series of minerals have been mentioned in relation to abortion, but a direct case report can be found only in case of iodine deficiency, with or without the common effect of thyreostatic compound in cruciferous and or nitrate in the drinking water. The iodine deficiency will decrease the intensity of the whole body's metabolism and finally will end in abortion. From the vitamins, the riboflavin, the B2 deficiency may cause early premature birth, approximately 2 weeks before term. The folic acid in human may cause spina bifida, premature birth, and also abortion. The biotin have a similar effect in mice. It is teratogenic, so to say, per se, and will cause abortion. The vitamin A is acting in gene expression of many essential genes. By this way, the vitamin A deficiency of pregnant sows may cause malformation of the aborted or newborn piglets. It's highly important to make a remark. The overdosage has the same effect by the toxic effect of the vitamin A. It is known that in human field there is a medical recommendation that pregnant women are recommended not eat more liver than once a week. In the general study plan, we are at the environmental and endogenous pollutants, toxins, heavy metals. And first we have to see the plants of the meadow for grazing animals. More and more breeding sow are kept in the meadow. Plants contain antinutritive substances, toxins, nitrate, and phytoestrogens, which plants are primary in provoking abortion. This is the locoweed. These are the piperidin containing plants like the poison hemlock or tobacco or juniper. And the NSP, the non-starch polysaccharide, for example, of many seeds, included the soybean seed, may contribute to the change of the microbiome and disturbance of the immune system and abortion. The industrial pollutants are very dangerous. The carbon tetrachloride, the trichloroethylene, and the benzene may cause spontaneous abortions both in human and in animals. The polychlorinated and polybrominated biphenyls, the PCBs and PBBs, were discovered by the Niagara Falls. They found in the 1960s that in some villages the abortion rate in human and even in domesticated animals increased. And the reason was the industrial pollution with PCBs and PBBs. The PCBs may have considered as families, and families of more than 100 particular chemical compounds, and they can be classified in estrogen-like PCBs, dioxin-like PCBs, and phenobarbital-like PCBs. And the first two groups are likely to cause abortion. The dioxin is itself a group of chemicals with cancerogenic, estrogen-like, and teratogenic characteristics. And all the three are able to provoke abortion. Back to the PCBs can cause a direct chromosome breakage, which is also a possible cause for abortions. The PCBs in mice will cause hydronephrosis of the fetuses, and the possible death of the fetus may cause a secondary abortion. The PCBs can be found in fish meal because by the underwater pollution it can get into the oceans and in the fish meat, fish body. But also the fatigue cooking oil, if you use many times, similarly it will be accumulated beside the PCBs, the dioxins. The oil cakes and its residues, the solvent meals like the soybean and fish meal, etc., may also contain PCB PCB traces. Heavy metal environmental pollutants from the traffic and from the industrial activity may cause mercury and lead contamination and consequent abortion. Pesticides and fungicides are also able to cause abortion. Technological origin has the following group of pollutants. Namely, they are related to the everyday technology in a big farm where they are using these, and in some accidental cases we have poisoning and related abortion. For example, crazole spray is used for eliminating mange and louse in an animal facility. And if the animal have access to it, they can get the poisoning. Or the decumarol, the anti-vitamin K substance, which is used against the rat invasion. And they really kill the rats after some intake, but the rat cadaver itself may be poisonous for the pigs. Of course, if the rat poison is available, this is even worse. So the decumarol will counteract the vitamin K and will cause disseminated bleeding everywhere in the body, including the uterus, and consequent abortion. Sometimes the facility heaters are not working properly and they will emit carbon monoxide. This carbon monoxide would be cause stillbirth and autolyzed full-term fetuses. It seems to be that the early fetuses are not so affected. After necropsy, you can find the fetal tissues are cherry red. Mycotoxins, as a pollutant or going on, are secondary metabolic product of the molds, and some other fungi. The first you have the whole list which are more or less related to the abortion: ergotism, the different molecules of aflatoxin, the F2 toxin or zearalenone, the fumonizins, the trichotessins, the T2, the DON, and the DAS, and newly the stachybotryotoxicosis, which is previously used to be a independent form of mycotoxicosis. So the first is maybe the classical form is the ergotism, which is an intoxication after ingestion of sclerotia of the parasitic fungi Claviceps purpurea, which are grown on feed, mostly on grain, but also on hay. They are producing ergot alkaloids, which has a potent oxytocic-like action. If fed pregnant sows, ergotized grains result in lack of udder development, with subsequent agalactia at parturition and the emaciation of the newborn, and the new piglet mortality will increase, but also may cause abortion or piglets born with the smaller body weight. The F2 toxin or zearalenone will cause the so-called estrogenism and vulvovaginitis mostly in pigs. The action of the mode of action of zearalenone is that zearalenone may bind to receptors for estradiol 17 beta, and this complex after that will bonds to estradiol sites on DNA, preventing the real estradiol 17 beta action. In this way, we can tell that clinically we will see a estrus-like state, but innerly, on the level of hormone production, nothing. What will happen? Rarely only comes the abortion, but the so-called estrogen syndrome in the newborn piglets with swollen genitalia and sway back and high mortality of the sows affected during the last third of the pregnancy. The fumonizins may cause acute pulmonary edema and hydrothorax in swine. Previously, it was called that disease of the fattening or fattening pig disease. But today we know that these are mycotoxins, and sows can also receive after having ingested the infected feedstuff. Sows will survive, but after recovery from the acute disease, they may show abortion 2-3 days later. The next group are the trichotessin structured mycotoxins, or fusariotoxicosis in the old name. The most important members of this group are the T2 toxin, the DON or vomitoxin, and the diacetylstirpenol. They are all immunosuppressive. DON, of course, will cause a feed rejection, but the most important, beside the immunosuppression, which is itself may cause abortion, inhibition in corpus luteum of the progesterone synthesis, and beside the neural side effects, seizures. Of course, all these factors may facilitate and contribute to an abortion. And finally, in this group, the previously treated as an independent disease, the stachybotryotoxicosis. This is an abortion by macrolytic trichotessin toxins, but with a lot of bleeding and very typical abortion in gestant sows. To make worse, even more worse, we have synergetic actions between other factors causing or facilitating abortion, and mycotoxins and industrial pollutant. For example, if you have a higher nitrate level in the drinking water, plus mycotoxins, or you have PCBs and mycotoxins in a relatively slow concentration in the drinking water, you can get this synergetic action. It will be demonstrated in fish model. You can see that aflatoxin B1 in a very, very low concentration. The concentration used to be 1 ppb parts per billion. And even it was able to cause local necrotic spots on the liver. The same concentration plus a small amount of cyclopropamide in the right picture will cause severe liver cancer. You see the tumors are overwhelming on this one. So the effect is not 2 plus 2, it's 4, but 2 plus 4 is 6, but 2 plus 4 are 8. So this is a synergetic effect. The detrimental effect on reproductive performance of pesticides are given and fungicides are given in a rat model. The effects of long-term treatments with diethyl dithiocarbamate on litter size, mortality, onset of puberty in form of age at vaginal opening, and gender ratio in the rat is the following. They control the saline injection. They used first in the first column M, first generation, maternal generation, experimentally virgin. And after TCF, they used females from previously treated mothers to see the transgenerational effect of the fungicide, of the chemicals. You can see that the first generation shows not differences to the control in the stillbirth, but the mortality increased, and owing to the treatment, the total mortality at weaning of the pups 44% compared to 14, and the age at vaginal opening increased by the treatment by 2-3 days. And what is interesting, that the female-male ratio, which shouldn't be 50-50 or 52 to 48 female males, absolutely changed, and mostly the females died because 1 to 2 is the ratio in the treated animals. And interestingly enough, the second generation was also treated and compared to saline treatment, physiological saline, and you can see that the stillbirth is very high owing to the treatment. The mortality is very high. It is 2/3 of the animals died. The age after vaginal opening or the puberty onset is later, 3 days, and even dramatic is the fall of the females, and the ratio of female to males is 1 to 3 instead of 1 to 1 or little bit more female than males. So you can see that the pesticides and fungicides shows detrimental reproductive effect, may cause stillbirth and abortion, and this is a transgenerational type of influence. and to see that how the immunological factors can contribute to the abortion, it should first overview that the immunology of the pregnancy is extremely complex. The fetus acting as graft in the uterus requires the presence of maternal blocking antibodies, which, along with the anti-inflammatory effect of progesterone, prevents the rejection of fetal tissues, which poses antigens that are quite different from those of maternal tissues. Okay, so you see that the maternal blocking antibodies are immunoglobulins which coat the foreign fetal antigens on the placenta. The way of action may lead to its deficiency in sows and may contribute to the abortion. The progesterone level of the mother's blood has an anti-inflammatory and calming, relaxing effect to maintain the pregnancy. An important immunological factor is the histocompatibility locus antigen, the HLA. The difference in maternal and fetal HLAs may account for the production of blocking antibodies. It is not known how the mode of action can contribute together with the waterborne pollutants and phytotoxins and mycotoxins to the abortion. But we know that the inbreeding effect is related to the genetics of the major histocompatibility locus antigen. And all the previous immune responses are modulated or compromised by the mentioned pollutants. The early pregnancy factor with immunosuppressive properties appears in the serum of 48 hours following fertilization. How this related to the abortion? Yes, the spontaneous abortion and early parturition, the stillbirths in swine, is preceded by a reduction in early pregnancy factor. Reduced immunosuppression may contribute to abortion. This is also similar in human. Allergic conditions can contribute to this topic, namely in the cereals. For example, in the cereals alpha amylase inhibitors and agglutinin in the germ of the barley, rice, and rye may cause allergic conditions contributing to the abortion. This is similar to the baker's asthma, which is triggered by the inhalation of the meal flour dust. And whoever visited a swine facility can remember that the feed make unfortunately dust. So this inhalation way is possible for the animals too. The second important group of immunological factors are the lectins or phytohemagglutinins, which can be found as anti-nutritive factors in the alfalfa meal and also in the soybean. Both are important components of the sow's feed mixture. And also the gluten in cereals, which is a mixture of proteins, which major allergic component is being the gliadin. And these lectin are competing for binding sites on the cortisol binding protein, which may result in elevated plasma cortisol level. And the high cortisol and the parallel low progesterone lead to bronchospasm and smooth muscle contraction. The common mechanism of all these allergic anaphylactoid responses is mediated by prostaglandin metabolism. See the introduction and the prostaglandin E2 surge, and this is related to the bad ratio of omega-3, omega-6 fatty acid intake. You know that the omega-3 fatty acids are anti-inflammatory, and the omega-6, which is unfortunately dominant, are pro-inflammatory fatty acids. And by this way, the COX system, the cycle oxygenase system, is affected, and it is pushed towards the direction of production of inflammatory prostaglandins and leukotrienes. And the allergic reaction is every time smooth muscle spasm, bronchospasm, uterus spasm, etc. The question arrived that the abortion may be prevented by feeding of omega-3 fatty acids and non-steroid anti-inflammatory drugs. The answer is yes, but the problem is how to schedule in a big animal facilities these substances. In the general study plan, we arrived to the amoeba parasites and algae-related problems. This is the so-called Legionella type model, showing that in some cases the real pathogen is living as a parasite in some amoeba-like amoeba organisms, and in this way they can be highly resistant. The air handling and watering systems of swine premises contribute to the accumulation of fungi, algae, microalgae, protozoa, and bacteria. And also not the human Legionella, but the Legionella-like amoebic pathogens, LLEP, are natural inhabitants of the water. And if the water sources are infected, they can survive. They are amoeba parasites. The pathogens harboring in amoeba are more resistant to various environmental stresses, such as the disinfectants that we are using for the disinfection of the water, drinking water. In the next, I will show you only as an illustration the most important harmful algal bloom species, which is not only in a free surface in a river or lake, but also in a water tank can be seen. You see, for example, microcystis cyanobacteria. And the global warming is contributing to this process. In the summertime, water tanks and towers can be turned into incubators. The corrosion products like the iron promote bacterial algal growth. And what is worse, the water chlorination practice using sodium hypochlorite decomposes toxins. Okay, but at the same time may cause toxin release from algae and the also toxic cancerogenic trihalomethane production. One more picture about the harmful algal bloom species and to see some details about the cyanotoxins, which is the most dangerous. From 40 species of cyanobacteria, we have many toxins, but four groups of toxins should be mentioned. The microcystins are as cyclic peptides, the cylindrospermopsin, tricyclic alkaloids, the anatoxin A, which is a bicyclic secondary amine, cholinergic agonist, potent neurotoxin, and the saxitoxins, which are carbamate alkaloid neurotoxins. And what are they doing? During the algal blooms, there is an accumulation of toxins, and it can be found in the plumbing systems, but also by different ways in the fish meals can be found, and they will cause porcine neuromuscular disorders. Reproductive problems included abortion, and the microcystins itself, produced by the microcystis aeruginosa, may cause hepatocellular damage, liver necrosis, and abortion. To final with, there is a list of potential infectious causes which should be excluded if you have abortion, and only after can you check the different etiology of non-infectious abortion. Finally, I summarized as a take-home message the whole lecture in form of brain map. You can overview, and to finish with, be optimistic, and thanks for your precious attention.